Hello everyone, welcome to ITLS Academy. Our today's lecture is Enumeration of Microorganisms. First of all, we will see what is enumeration of microorganism. What does it means? So the very first point is enumeration in microbiology is the determination of the number of individual viable microbes in a sample. Here, four basic techniques are possible. The most commonly used forms of bacteriological analysis in food microbiology are detection and enumeration. Next point is the presence of specific bacteria and their concentration must be determined to assess and control safety hazards. The potential for spoilage or to ensure correct product characteristics. Microbial growth is essentially synonymous with microbial reproduction. As we know that microorganisms grow by the process of cell division and they reproduce also by the process of cell division. So growth and reproduction are synonymous in the case of microorganisms. To determine rates of microbial growth and death, it is necessary to enumerate microorganisms, that is, to determine their numbers. It is also often essential to determine the number of microorganisms in a given sample. Next point is the most common procedure for the enumeration of bacteria is the viable plate count. In this method, serial dilution of a sample containing viable microorganisms are plated onto a suitable growth medium. It is assumed that each bacterial colony arises from an individual cell that has undergone cell division. So, here is the definition of enumeration. Enumeration is counting of microorganisms, and these are present in a sample. You have to count microorganisms if you are given a sample, and if these are colonies of microorganisms, then you just have to count what number of organisms are present in this particular sample, and this counting is known as enumeration. Enumeration is done to know the intents of presence of the spoilers in the spoiled food. Most of the time it is used if some microorganisms are present in any food, then for the what amount and what kind of organisms are present, to determine it, you have to do enumeration for the microorganisms. To detect which type of organism is responsible for the spoilage, You can also determine whether what kind of spoilage that particular organism is responsible to do. Mostly it is done two important methods. The methods for the enumerations are vival count and total count. In the vival count, you have to count the vival microorganisms, while in the case of total count, the counting is done or the total sample is counted. So now we will see what are the objectives of this test, why we do enumeration of microorganisms. After completing this exercise, the student will be able to perform the following objectives. Very first objective is state the formula for determining the number of CFUs per ml of sample when using the plate count technique. Point 2. When given a diagram of a plate count dilution and the number of colonies on the resulting plates, choose the correct plate for counting. Determine the dilution factor of that plate and calculate the number of CFUs per ml in the original sample. Next point, state the function of a spectrophotometer. Remember, these are the objectives of the test that we are going to do, that is enumeration of the microorganisms. Next objective of the test is state the relationship between absorbance, that is optical density, and the number of bacteria in a broth sample. And the next one objective is state the relationship between percent light transmitted and the number of bacteria in a broth sample. So here comes procedure for the enumeration. How we can count microorganisms. So the procedure involves perform a serial dilution of bacterial sample according to instructions in the lab manual and plate out samples of each dilution using the spin plate technique. For the results, using a colony counter. Most of the time this is something like this. This is divided in 4 parts and you just have to count colony in all of these divided parts. In 4 parts you will count the colonies and finally divide with multiply with dilution factor, and by the formula you will found the number of microorganisms. So by use of a colony counter, count the number of colonies on a plate showing between 30 and 300 colonies. And by knowing the dilution of this plate, calculate the number of CFUs per ml in the original sample. Let's say discussion. As part of daily routine, the laboratory microbiologist often has to determine the number of bacteria in a given sample, as well as having to compare the amount of bacterial growth under various conditions. We can say that enumeration of microorganisms is especially important in dairy microbiology, food microbiology, and water microbiology. And also, the number of colonies can be used as a measure of the number of viable cells, or we can say these are the measure of number of living cells in that particular known dilution. So in this diagram, the process of dilution is shown. You can see this is the sample of E. coli is taken in this flask. 1 ml from this sample is transferred in another tube, and then another tube, this concentration is 10 to the power minus 1, minus 2 and so on. Continuously transfer of 1 ml. Although we have discussed the process of serial dilution in detail in our previous lecture, you can refer to that lecture, but I am also showing again this process of serial dilution in this lecture, but in detail we have discussed in previous lecture. You can see these those details. The transfer of 1 ml from this concentration to this and from this to this. This process continue till the last concentration we get. And this is the method of plate count method or viable count method. When you will get this final concentration, you have to pour it on that particular counting meter and you have to count the cell number. Next method is direct microscopic method. This is the method in which total cells are counted. Let's see direct microscopic method. In the direct microscopic count, a counting chamber consisting of a ruled slide and a cover slip is employed. It is constructed in such a manner that a known volume is delimited by the cover slip, slide and ruled lines. This is the diagram showing the total count method. In this method, the number of bacteria in a small known volume is directly counted microscopically, and the number of bacteria in the large original sample is determined by extrapolation. Another term is turbidity. When you mix the bacteria growing in a liquid medium, the culture appears turbid. Turbidity is a process, is a phenomena in which if there is a liquid medium is present, and you pour cells in this medium, then it will look like something like a turbid, or we can say it will have less visibility. So this is known as turbidity. This happens because a bacterial culture acts as a colloidal suspension. Here bacterial culture will act as a colloidal suspension and which blocks and reflects light passing through the culture. This colloidal suspension blocks the reflecting light. That's why the visibility becomes less and the medium known as turbid. Within limits, the light absorbed by the bacterial suspension will be directly proportional to the concentration of cells in the culture. By measuring the amount of light absorbed by a bacterial suspension, one can estimate and compare the number of bacteria present in that sample. The instrument used to measure turbidity is a spectrophotometer. This is an instrument which consists of a light source, a filter which allows only a single wavelength of light to pass through, the sample tube containing the bacterial suspension, and a photocell that compares the amount of light coming through the tube with the total light entering the tube. So, the percent of light transmitted is inversely proportional to the bacterial concentration. The absorbance of optical density is directly proportional to the cell concentration. These are two important points to remember. You must have to remember this while the counting methods. Here comes the procedure which test we are studying. That procedure is given over here. Let's discuss this procedure in detail. First is plate count and it is to be done in pairs. First point is take 6 dilution tubes, each containing 9 ml of sterile saline. Aseptically dilute 1 ml of a sample of E. coli. This dilution will include these few steps. So let's see these steps. Point A is remove a sterile 1 ml pipette from the bag. Do not touch the portion of the pipette that will go into tubes and do not lay the pipette down. These are the precautions whenever you are doing experiments. From the tip of the pipette to the zero line is 1 ml. If this is your pipette, then this is if it is 0, if this is tip and this is 0 line, then this is 1 ml. Each number division, that is 0.1, 0.2, etc. represents 0.1 ml and each division between two numbers represent 0.01 ml. Now, insert the cotton tipped end of the pipette into a blue 2 ml pipette filler. Next is flame the sample flask. Insert the pipette to the bottom of the flask and withdraw 1 ml up to the zero line of the sample by turning the filler knob towards you. Now draw the sample up slowly so that it isn't accidentally drawn into the filler itself. Now reflame and cap the sample. Next point, flame the first dilution tube and dispense the 1 ml of sample into the tube by turning the filler knob away from you. Now draw the liquid up and down in the pipette several times to rinse the pipette and help mix. Now reflame and cap the tube. Next point, mix the tube thoroughly by holding the tube in one hand and vigorously tapping the bottom with the other hand. This is to assure an even distribution of the bacteria throughout the liquid. Next point, using the same procedure, aseptically withdraw 1 ml from the first dilution tube and dispense into the second dilution tube. Continue doing this from discard the pipette in the disinfectant container on the bench top. Although we have also seen this whole procedure in our previous slide, I can show you. This is the process that we are discussing in point 1. You just have to take your pipette, dip it into the sample and take 1 ml, transfer it in this tube and then this process goes on. So we have taken our 1 ml of sample. Now next one is these pipetting and mixing techniques will be demonstrated by your instructor. So the next point, that is second point, is using a new 1 ml pipette, aseptically transfer 0.1 ml from each of the last 3 dilution tubes onto the surface of the corresponding plates of trypticase soy agar. Let's see this process. This is the process. You just have to take the last 3 concentrations and draw 0.1 ml from each of the tube on the soy agar plate. Third point is using a turntable and a sterile bent glass rod, immediately spread the solution over the surface of the plates as follows. The process for the spreading is given in these steps. Let's see. Point 1 is place the plate containing the 0.1 ml of dilution on a turntable. Point B, sterilize the glass rod by dipping the bent portion in a dish of alcohol and igniting the alcohol with the flame from your burner. Let the flame burn out. Point C is place the bent portion of the glass rod on the agar surface and spin the turntable for about 30 seconds to distribute the 0.1 ml of dilution evenly over the entire agar surface. Point D is replace the lid and re-sterilize the glass rod with alcohol and flaming. and the last step is repeat for each plate. This process will be repeated for each plate. Next one, discard the pipette in the disinfectant container. These 6 points are given with regards to spreading of the solution over agar plate. Our fourth point is incubate the 3 agar plates upside down at 37 degrees centigrade until the next lab period. Place the used dilution tubes in the disposal basket in the hood. Next is direct microscopic method. Point 1 is pipette 1 ml of the sample of E. coli into a tube containing 1 ml of the dye, and the dye is methylene blue. This gives a 1/2 dilution of the sample. Next point, using a pasture pipette, fill the chamber of a Peterhof hose containing chamber with this 1/2 dilution. Third point, place a cover slip over the chamber and focus on squares using 400 or you can say 400 times magnifying objectives. Point 4 is count the number of bacteria in 5 large double line squares. For those organisms on the lines, count those on the left and upper lines, but not those on the right and lower lines. Divide this total number by 5 to find the average number of bacteria per large square. Now in the last, calculate the number of bacteria per cc. Third is turbidity. Your instructor will set up a spectrophotometer demonstration illustrating that the number of bacteria in a broth culture increases, the absorbance increases. Now come to the results. First of all, we will see results for the plate count. Number 1. Choose a plate that appears to have between 30 and 300 colonies. For the sample 1 upon 1 lakh dilution plate. Next is sample 1 upon 10 lakh dilution plates. And the third sample 1 upon 100 lakh dilution plates. Count the exact number of colonies on that plate using the colony counter as demonstrated by your instructor. Calculate the number of CFUs per ml of original sample, and this is the formula for the counting of CFUs. So the number of CFUs per ml of sample is equal to number of colonies, that is the colonies between 30 to 300 plates, into the dilution factor of the plate counted. Results for the direct microscopic methods will be: observe the demonstration of the Peterhof Hauser counting chamber. For the turbidity, observe your instructor's demonstration of the spectrophotometer. This is how we can calculate results of plate count, direct microscopic method and turbidity. So thank you very much guys for watching this session. Follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn. And for latest videos and updates, you can subscribe our YouTube channel, that is ITLS Academy. Thank you very much for watching.